All right, we're going to do Chapter 11, Part 2, Notes, which includes Section 11.4 in your textbook. We're going to do this in a flipped format to save class time, and you can read about these disorders further in your textbook. So we're going to talk about, in this presentation, human genetic disorders. So patterns of inheritance, you need to remember that we have autosomal chromosomes, which are chromosomes 1 through 22. So we're excluding the sex chromosomes. For nomenclature purposes, the X and the Y. And when you have an autosomal recessive disorder, we're showing that we have the homologous partners. So you need to get two bad copies in order to show that disorder, versus autosomal dominant. In that particular case, that mutation, that disorder is dominant, so you only need one copy of that genetic disorder and you actually show or express that particular disorder. So another type of inheritance pattern is called sex-linked, and sex-linked is when you find that particular gene is located on the sex chromosomes. Generally speaking, it's on the X. And females have an advantage here because, of course, females have two X chromosomes and males only have one. So females can be homo or heterozygous for that particular genetic disorder, where males, they're hemizygous, meaning they only have one possible chromosome that they can get that good or bad copy of that disorder. So we generally see this expressed more in males than females, and we will talk about this in more detail in Chapter 12. So in this section, we're going to talk about genetic disorders. We're going to focus on the autosomal recessive, and with that type of genetic disorder, we're going to talk about Tay-Sachs, cystic fibrosis, and PKU. Now, the other type of genetic disorder we're going to talk about in this section is autosomal dominant, and we'll talk about neurofibromatosis, achondroplasia, and Huntington. So the first autosomal recessive disorder that we're going to talk about is Tay-Sachs disease. We find the gene for this disorder is particularly high in the Jewish community, and this has to do with their policy in relationships, you can say, and that they try to marry only people within that same Jewish sector. Of course, there's different branches within the Jewish community. But what we find is because they do not go out and mingle their genes, we could say, with the regular population, this gene has gotten into that genetic pool and it's kind of stuck there. So we'll see a lot more carriers within this group than we will outside, and because you have a higher number of carriers, that increases the potential of their offspring being born with Tay-Sachs. This particular disorder involves an enzyme called hexoaminidase A, and it breaks down this glycolipid which accumulates in the cells and impairs function. This glycolipid is found in high concentration in brain cells. So what happens is a child is born with the disorder, and the child may look fine and may start doing the normal things that babies do, rolling over, sitting up, cooing, and then as this glycolipid accumulates more and more in the brain, it actually starts to impair the proper nervous function, and development will actually stop and then reverse itself till eventually the child is just kind of in a vegetative state and then will not be able to breathe and eat, and it will die. This is a disorder that does lead in death. So in this slide, you can see that on the top, that's a normal developing neuron. And here on the bottom, we have a neuron that is filling up with that ganglioside and will stop working properly. You can also see the lysosomes and how they're nice and healthy looking, and here they're bulging because they're trying to break down that extra material that the cell's producing, and they just can't keep up. In this slide, what I'm trying to show is that you could have two parents that are perfectly healthy, and they don't have this gene expressed, but they could both be carriers. And when they're both carriers, there is a possibility that their children could end up being homozygous recessive. So they're kind of showing you here the possible gamete combination. Another way of demonstrating this would be a Punnett square, but we're going to get the same outcome, and that is 25% of the offspring will be little r homozygous recessive. 50% will be heterozygous, which means they potentially could carry that. Well, they do carry that, but they potentially could have children with the disorder if, again, their spouse also is a carrier. And then we have 25% that are homozygous dominant, which means not only do they not have the disorder, but their children will not have the potential of having this disorder. So another example of this autosomal recessive disorder in humans is cystic fibrosis. Cystic fibrosis is a genetic disorder which makes a faulty chloride ion channel in the cell membrane. This results in the inability to maintain homeostasis in specific tissues. Those tissues include the lungs, digestive system, and the endocrine system. So CF symptoms include very salty sweat due to this excess chloride ion, since the protein that's carrying or moving that particular ion across the cell membrane is not functioning properly. It also causes problems with mucus formation. We learned that in our osmosis diffusion chapter, I believe that was chapter 5, that water will tend to follow the solute in order to kind of dilute it out. So in this case, water will tend to follow that excess salt in CF, which causes mucus to form inside the lungs, which then prevents proper gas exchange. We're also going to get mucus that will clog the pancreatic duct, which prevents the digestive enzymes from getting into the small intestines. There was that common bile duct we talked about freshman year that goes into the first part of the small intestine called the duodenum. Many of those digestive enzymes are produced in the pancreas, so without those enzymes being able to get into the duodenum, people with CF have a hard time breaking down their food properly and absorbing the nutrients that they need from their food. So to counter that, they'll take a lot of different enzymes before they eat. All right, the treatment in CF includes physical therapy to help dislodge the mucus from the lungs. They'll do this 2 and 3 times a day. And as I mentioned in the last slide, they will take digestive enzymes before they eat any food. So the average life expectancy of a child with cystic fibrosis used to be 10, 14 years tops. With today's treatments, though, patients can live up until their mid-30s, and hopefully with more technology, even longer. We have found the gene for cystic fibrosis. There is an error in the DNA. It is on chromosome 7, and what we found with this gene is just one nucleotide has been changed, and that one nucleotide change has altered one amino acid, and that one amino acid has prevented that protein from folding properly. Therefore, that protein doesn't work. Some of the advances in cystic fibrosis include gene therapy. They're trying to put a good copy of that gene into the cells of those patients that have CF. We're experimenting with using viral hosts as a delivery system, because that's the biggest problem, is how do you deliver that good gene? So we're using viruses to do that. Phenylketonuria, often called PKU because it's much easier to say, is there's an enzyme that we have that will break down the amino acid phenylalanine. The enzyme, the instructions for this enzyme is located on chromosome 12. Now, if you have two bad copies of this enzyme, then what happens is the phenylalanine will then accumulate into toxic levels in your body, and it tends to affect the nervous system. So if we can identify individuals that are homozygous recessive for PKU, then we can put them on a diet that is free of phenylalanine and development will occur normal. However, if we give them foods that contain the amino acid phenylalanine, it can cause some developmental problems. All right, now we're going to talk about autosomal dominant disorders. Now, the difference between an autosomal dominant is that you only need one copy of the bad gene and it's expressed. You see it. So where the autosomal recessives, the heterozygous, were still normal, with the autosomal dominant, the heterozygous has a disorder. So one example of an autosomal dominant disorder is neurofibromatosis that is found on chromosome 17, and it interferes with the cell cycle. It's found in the skin and nerves, and the result of this disorder is large tumors. So here's a pedigree with a human dominant disorder. Remember, with dominant disorders, you only need one copy of this gene to actually show the disease. So when we look at this first generation, we have this individual in red. The red will be the individual that has a disorder. The person with the red square, so the grandfather here, could be, let's say, big H, or big H, little h. Now, the mom is little h, little h. We know this because she doesn't have the disorder, and this is the only way you will not have the disorder. So you can kind of go through, and you know just all the blue square and circles are going to be little h, little h. It's the only way they can be normal. Now, let's look at the afflicted, the individuals that have it. Now, like we said, this grandfather could be big H, or big H, little h, but when we look at his children, the only way they can have normal children is if he is a carrier for the normal gene. So we know anybody that's red here is going to have a big H. The question is, what's their second gene? Now, we know in the second generation that all of the offspring are going to have a little h from mom. When we go to the third generation, it might get a little more confusing. This one's going to have a little h from mom. This individual will have a little h because of dad. Little h because of dad. So we could, in this pedigree, we're actually able to fill out all the genotypes. You just need to remember that dominant disorders, you could have one bad copy or two bad copies. It doesn't matter because it's dominant. You're going to see it. Now, the one thing that is different between these two individuals is that this one, all his offspring will have the disorder. This individual, only half will have it. So that is the difference between the homozygous dominant and heterozygous. An example of a dominant disorder is Huntington's disease. Now, with Huntington's disease, the symptoms are not generally seen until middle age. So the problem is, by the time the person who has this disorder realizes they have it, they've generally already had children, which is how this disorder gets passed on. Death generally occurs 10 to 15 years after the symptoms start. So my question here is, are these children's carriers? Well, again, if we're looking at autosomal dominance, mom here is normal. Dad's the one that's affected. And we can say if he does have Huntington, so he's got big H, little h. The children, these are the possible combinations for children. We have 2 here that actually have the gene for Huntington. So they are going to actually have Huntington's. So the question is, are they carriers? Well, you really can't be a carrier for an autosomal dominant disorder because the term carrier implies that you carry the gene but don't express it. All these individuals that have just one copy are going to express it. If anything, you can say that they are a carrier for the normal gene. So they do have the potential of having normal children, meaning children without the disorder. An example of a human dominant disorder is achondroplasia. This is a very common form of dwarfism. It affects the growth in the long bones, so they have shorter arms and legs. The disorder has been identified as a faulty gene located on chromosome 4. There are 3 genotypes then, big A, which that is a lethal combination, so generally they don't carry those the offspring that are big A to term, so they don't survive. So the dwarfs that we actually see are heterozygous. They're big A, little a, and again, remember this is dominant, so you just need one big A, and you express that disorder. And then we have little a, which is normal. In this picture, these are actually twin brothers, not identical, but paternal brothers, and you can see the one ended up getting, this one here, the young boy is big A, where his brother is little a, so he's normal. So here's a pedigree of a family with achondroplasia, and you can see here that the father is a dwarf, mom is normal. They have 1, 2, 3, 4 children. Of the 4 children, they actually have 2 which are normal and 2 that are dwarves. So these 2 here are dwarves. These 2 here are normal. And you can see how the individuals that are normal, they don't have any dwarves in their offspring, and that makes sense because in order to be normal, you only get little a. However, if we look here with the 2 that are dwarves, they actually do have some dwarfism in their children. Because again, they have that little a, big A, so it's that big A that they're passing on, and since it's a dominant disorder, that it's being expressed in a couple of their offspring. So we see in dwarfism, in this particular case, in all 3 generations. But we also see some normal individuals as well. So that concludes the flipped classroom presentation on human disorders. If you'd like to talk further on these topics, please bring it up in class. If not, hopefully between the lecture and the book, you understand the idea of autosomal recessive and autosomal dominant disorders